Hi and hello, welcome to online biology class because of Ideal Others Educational Institution and the Your. I am Sadish Kumar going to present the topic in the field of organization of tissue. So the organization of tissue is a ninth standard biological science. This is unit number 18. So the continuation of the organization of tissue from we will see today's topic regarding meiosis. So what is meiosis? We know very well. The last class I give you some kind of basic information and introduction regarding the meiosis. So meiosis is a type of cell division. So we know very well a single cell is get divided into two cells. That is a simple mechanism for cell division. So the cell divisions are three types. One is called amitosis, mitosis and meiosis. So amitosis, which is commonly takes place on unicellular organism. Then meiosis and mitosis both are takes place on multicellular organism. So mitosis, which is takes place on the body cell, and by the same time the meiosis was takes place on the gonads or reproductive cell. Then the mitosis is also called an indirect division, and meiosis is also called an reduction division. So why we are called upon it is a reduction division? Because they are reduced their own chromosome number into half because of in meiosis 1. So it is called an reduction division. So let us enter into this topic meiosis. Then the term meiosis was coined by a farmer in the year of 1905. One more question. It is the kind of cell division produced by the sex cells or gonads, which is takes place on the oogamy and spermatogamy, and by the same time they are producing the sperm cell and egg cell. So this is the sex cell or gonads. It is also called an reduction division. One more question. Because why it is called upon reduction division? Because the chromosome number is reduced to haploid n from diploid. So diploid is a 2n, haploid is a single n. So they are reduced into half. Two nucleus is divided into one nucleus. So this is called a reduction division. And a meiosis produces 4 daughter cells in a parent cell. Then meiosis consists of 2 divisions. So what are the divisions you will see later. Then they are produced by the 2 cell. Then the 2 cells are divided into 4 haploid single cell. The end product of meiosis, they are produced by 4 haploid number of daughter cell from the parent cell. And the two types, one is called heterotrophic division and another one is called homotrophic division. First division and second division. Hetero means different. So different kinds of processes takes place. Homo means same type of division. So heterotypic division and a homotypic division. What are the divisions of meiosis? 2 more question. Heterotrophic division and homotypic division. Then what is heterotypic division? So it is a diploid cell into 2 haploid cell. So one single diploid cell 2n, they are produced with 2 single n containing haploid cell. The daughter cells residing from this division are different from the parent cell, the chromosome number. So it is called heterotrophic. From the parent to the daughter cell, they are having some kind of variation in the chromosomal number. So it is called an heterotrophic. Presence of variation and a difference, it is called upon heterotrophic. And second one, this is consist of 5 stages. So these are the things are seen about mitosis also. Prophase 1, metaphase 1, anaphase 1, telophase 1 and cytokinesis 1. So these are the 5 stages which is takes place on heterotypic division. Then prophase. So it is a longer duration and a subdivision of 5 divisions. So prophase is a longer duration for meiosis 1 and by the same time they are having a 5 subdivisions. One is called leptotin, zygotin, pachytin, diplotin and diakinase. Simple understanding, le sa padida. Leptotin le, zygotin sa, pa pachytin, D diplotin, da diakinase. So lesa padida. It is a simple identification of the remembering the subdivisions of prophase 1. Again I told you Lesa Padida. Lesa Padida. And this is the diagrammatic representation of prophase 1. So the spindle formation and the synaptid, a homologous pair of chromosome 2n. So they are consist of 4 haploid number of chromosomes. And leptotin, the chromosomes become uncoiled and assume a long thread-like structure and take off to a specific orientation the side nucleus. They form a Burnett stage. So we will see that the uncoiled, that is like in the form of idiaphum. So this is called uncoiled like structure, and inside of this nucleus, so everything takes place inside of the nucleus. And zygote outgoing, the two homologous chromosome approach each other, begin the pair. So the each and every type of chromosome, they are produced their own pair. So that is called a formation of homologous chromosome. This is called a synapsis, a condensed. So this is called a synapsis. And this is the diagrammatic representation how the formation of leptotin and zygotin. Then asters are formed and nuclei are developed inside of the nucleolus is called a leptotin. And zygotin, homologous chromosomes are formed. So orange color and blue color, pink color, violet color. So they are given for a bivalent. Bi means two. Two set of chromosomes are formed. So that is called a zygotin. And we will see about patches. That is a thick. The chromosomes are visible and long padded twisted thread. That is in the form of twist like a rope. The pairs are formed is called bivalent. The ropes are formed with two unequal and equal length. Then finally they are made down with one lobe. So it is called bivalent. Each bivalent now they are consist of 4 chromatids. They are make the cut and they are divided into 4 stages. And homologous chromosomes, each pair belongs to being two separate. So the homologous chromosomes are separated and they do not completely separate but remain attached together one or more points by X-shaped. We know that the structure of chromosome, they are always X-shaped. The center they are consist of centromere. This is called an chiasmata. So one more question, another one more question, chiasmata. And the chromatids breaks to this point and broken a segment may be interchanged. So the crossing over result, the genetic recombination is takes place. A crossing with the 2 different characters. So the genetic variation behind the stage. And diplotin. So this is the 4th stage. It involves the chromosome and each bivalent begin to split longitudinally, so horizontal, and into the similar chromatids. A homologous chromosomes repel each other and separate. Once they are make the cut, then they are separated and divided into opposite poles. And the chiasmata begin to move along the length of chromosome of the centromere towards the end of resulting the terminalization. So unwanted characters are eliminated and recovered characters are passed on with the next generation. The process is called an terminalization. Then this is the diagrammatic representation of patchotin, the formation of bivalent, and diplotin, formation of chiasmata, and diakinase, what it will be happen, the centromere and the microtubules are attached and mitotic spindles are appear, then they can move on with the opposite direction. They are ready for my metaphase. And the last phase we will see about the metaphase. The chromosomes moves towards the equator and finally orient themselves in the equator. The 2 chromatids of the each chromosome it do not separate. The centromere does not divide. So we will see that then black color line is given in the middle. The centromere is not divided and the metaphase plate is not divided, but they are having some kind of contact with their own offspring. And next one we will see about anaphase 1. Each homologous chromosome with the 2 chromatids and undivided centromere move towards the opposite pole. One is called upper side, another one is called down side. This stage of the chromosome is called an diet. So this stage is called an diet. So we can see that the meiosis 1, separation of homologous pair from the anaphase 1, they are attached with the opposite direction and they are pulled with by means of a spindle fibers. Then the telophase, a haploid number of chromosomes after reaching the respective pole become uncoiled and elongated. The nuclear membrane and nucleus are reappear to two daughter nuclei are formed. From meiosis 1, the formation of only a two daughter nuclei. Then we will see that there is a cleavage flow and a reforming of nuclei, that is called rearrangement of nucleus and the nuclear membrane, formation of two nuclei daughter cell. And final stage, that is called and cytokinesis 1. A cytoplasm is occur with the two haploid cells are formed. So the cytoplasm also undergo for division and the separation of two new daughter cell. And this is the overall representation of meiosis 1, prophase 1, metaphase 1, anaphase 1 and telophase 1, which can be give a very good representation, and what are the changes behind that is given from the diagram. And we can move on to the homotypic division. So second type is otherwise called as meiosis 2. Is otherwise called as meiosis 2. In this division, two haploid cells formed during the first meiotic division divided into four haploid cells. A one cell get divided into two cells, that is a meiosis 1. Then two meiotic daughter cells are converted into four haploid cell, is called a homotypic division or meiosis 2. And the daughter cells are similar to the parent cell in the chromosome number, so homotypic. So they are having a 46 chromosome, means the daughter cell also consists of 46 chromosome. It consists of 5 stages. Number 1, prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2. Then prophase 2, the centrioles divided into two, each one moved to the opposite pole. Asters and spindle fibers upper, nuclear membrane and nucleolus are disappear. So two stages are involved. Then formation of spindle fibers and the membranes are disappear. Second one, metaphase 2, the chromosomes get arranged on the equator, the two chromatids are separated. So we will see that the formation of centromere and chromatids available on the center position, they are going ready to separate. And anaphase 2, the separated chromatids become a daughter chromosome and move to the opposite. Now we can see that two chromosomes, they are given this diagram, separation of sister chromatids, they are separated and move on to the opposite pole by means of a spindle fibers. And telophase 2, a daughter chromosomes are centered and nuclear membrane nucleolus are appear. Now which is move on to the four haploid daughter cells, which is move on to the opposite direction. Nucleus and nuclear membrane are reappear and they are get ready for cytokinesis. Now we can see about cytokinesis 2. The two cells are formed from the each haploid daughter cells, resulting the formation of four cells haploid number with the chromosome. So what is the end product of meiosis 2? They are produced by four haploid numbers of chromosome. Four haploid number containing chromosomes are produced by meiosis 2. And this is the diagrammatic representation. After meiosis 2, we are get down with a 4 number of haploid cells. Then this is the overall representation of the meiosis process and meiosis 2. Then telophase, then followed by prophase 2, metaphase 2, anaphase 2 and telophase and cytokinesis are given this diagram. And this is the overall schematic representation of meiosis process, which is included by both meiosis 1 and meiosis 2. And what are the significance of meiosis? The constant number of chromosomes are given, the species is maintained by the meiotic division. During the meiotic, the chromosome number from the human being, ourselves, we are having a 46 number of chromosome. Our son and daughter also having a 46 number of chromosome, and grandson also consists of same number of chromosome. And then we will see that the difference between mitosis and meiosis. Mitosis occur in the somatic cell and meiosis occur on the reproductive cell. Then mitosis involves the growth and occurs continuously throughout the life, but meiosis occur only at the time of reproductive age. Then mitosis consists of single division and meiosis consists of two division, and two haploid daughter cells are formed in mitosis and 4 haploid daughter cells are formed in meiosis. And from the mitosis, the chromosome number and daughter cells are similar in the parent. They are always in diploid. In the meiosis, the chromosome number of the daughter cell just half, singly and haploid to the parent cell. The identical daughter cells are formed from the mitosis. The daughter cells are not similar to the parent cell and randomly assorted with meiosis. So up to that, the important question is difference between meiosis and mitosis, which is asked from the examination point of view. We are successfully completed for unit number 18. We are having a one more lesson for your quarterly examination, that is a plant physiology. We can start the particular topic from the next class itself. And then bye bye and thank you very much from Sadish Kumar from Biological Science Department. Thank you one and all.